When we look at osmosis, osmosis is going to be the diffusion of water. So when we look at osmosis, it's the diffusion of water from an area of high concentration to an area of low concentration through a semi-permeable membrane. So when we look at a beaker of water, we can put a semi-permeable membrane. So right here is our semi-permeable membrane. We'll fill it with some water, and on one half the water, we're going to fill it with some molecules. So it could be sugars, could be salts. We're going to put some solutes on one side of the membrane. And now when we look at this, we talk about the membrane as being semi-permeable. So if you watch this little solute, you'll see that it can't cross through the membrane. But water is free to move in both directions. So when we look at this beaker here, and we look at, well, which side has a high concentration of water? Because osmosis is flowing from high to low. Well, on the right side here, we have 100% water. Where on this, if we have 15% solutes and 85% water, well, then you would see that water is going to move from high concentration to low concentration. So which arrow, A or B, is showing us the direction water is going to move? Yeah, B. When we look at it, water is going to move from 100% towards the area where there's less. So it's going to move from high concentration to an area of low concentration. And as the water moves in this direction, the beaker will actually fill on one side of that membrane. So water will move from an area of high concentration to an area of low concentration. So here, not as good, but you can kind of see how this would happen. Now, a common way to look at this in biology is with a U-shaped tube, where in the middle they put a semi-permeable membrane. And you can kind of see that on this side, there's more water, higher water concentration on the left, and less water on the right. So water flows from high to low, and the tube will actually fill with water. So over time, one side will actually rise up, and that will happen until the water is equal on both sides. If we watch, I don't know if, oh, maybe this is going to work. If we watch a quick YouTube video, you can see osmosis happen. So on the left-hand side here, there's sugar or sucrose solution. And you can kind of see on the right-hand side how that actually is filling up. Okay. All right. So that's osmosis in a U-shaped tube. Okay, so when we look here, let's have our semi-permeable membrane. Oh, shoot.
Hi, it's Mr. Anderson, and today I'm...
Okay, so you can see here we have our semi-permeable membrane and two sides, side A and side B. So when we look here, we call anything that is not water a solute. So this sugar molecule, this glucose, is called a solute. And if you look at it, you could see, if we think about it, which side, side A or side B, has a higher concentration of water, that's going to be side B. When we look at, well, which side has more solutes? Obviously, that's side A. So when we think about this, well, which way is the water going to flow? Is it going to flow into side B, or is it going to flow into side A? Well, water always flows from where there's a higher concentration of water to where there's a lower concentration of water. So the water is actually going to move into side A from a high concentration to a low concentration. So when we look at how we would explain this using like science words, we have three words. We have hypertonic. Hypertonic means more solute, less water, or a low concentration of water. So if we were comparing side A and side B right here, side A would be considered side B would be considered hypertonic compared to side A because there's more solutes and less water. Then if we look at the word hypotonic, hypotonic means less solute, more water. So that would be a high concentration of water. So in this example, side A is hypotonic, having more water when compared to side B. Now, if they're equal, that's going to be isotonic. So in this situation, water is going to move from where there's a higher concentration to where there's a lower concentration. It's going to move from the hypotonic side into the hypertonic side. So let's go ahead and see this in action. So here I have some water, and I'm going to add a red blood cell. Now let's pretend inside the cell there's two solutes, or two things that are basically not water. This could be organelles, salts, sugars, proteins, etc. Now if we put it into a solution, though, that has a whole lot of solutes, I want you to think for a minute, which is hypertonic? Is it the solution or is it the cell? Which area has more solutes and less water? In this situation, the solution is hypertonic when compared to the cell. It has more solute and less water in comparison. Now the cell, how would you describe the cell compared to the solution? And here, the cell is going to be hypotonic to the solution. There's less solute and more water. So we think, okay, well, water is going to move from high concentration to low concentration. So if I look at this, well, I know that inside the cell is going to actually have a higher concentration of water compared to the solution. And so water is going to flow from high to low. So if you think, well, what does that mean for the cell? Well, if we see, here's our water, and water is actually going to leave the cell. So the water is going to flow out of the cell, and the cell is going to lose volume. It's going to lose water, so the cell actually ends up shrinking. So if we do that again, we can see here, if the cell is placed into a hypertonic solution, what's going to happen is the water is going to move from inside of the cell where there's a higher concentration to where there's lower, and that's going to cause the cell to shrink. Now, what we can see here in real life, if you have strep throat is caused by bacteria. A bacterium is a single cell, and let's just pretend it's like 90% water. So here, if like growing up, I was always told gargle with salt water when you have a sore throat. Well, here's why. If I add some salt to some water, well, now that might be about 70% water, 30% salt. So if I go to gargle and here's my salt water, and the bacteria is growing in my throat, so now the bacteria that's 90% water, I can look and here this has a high concentration, and this has a lower concentration of water. Now let's think, which one's hypertonic? The water or the bacterium? Remember, hypertonic means more solute and less water. So in this situation, the one with more solute and less water is the salt water. So water is going to flow from where there's a high concentration to where there's a low. It's going to flow from 90 towards 70. So the water, is actually going to leave this bacteria. And well, what does that mean for the bacterium? Well, it's going to shrink. And now it's dead. And this is why gargling with salt water will help to prevent or at least slow down the growth of bacteria in your sore throat. Now, if we look at a couple other examples, here's a cell. And it's 98% water inside, 80% water outside. So, which area is hypertonic? Good, it's going to be the outside. The outside is hypertonic compared to the inside. Water is going to flow from high to low, and that cell is going to actually shrink. Now, if we have 98% water equal, the water actually still flows. It just flows in equal directions or equal rates inside and outside. So this is said to be isotonic. And then let's pretend we put a cell into 100% water. Well, if it's in 100% water, here's more water, here's less. That water is actually going to flow into the cell. So the water flows in, and if you can imagine, if that's gaining water, it's going to get bigger. Now, when we look at this, which area was hypertonic? Actually, the inside of the cell is hypertonic, has less water and more solutes. Area that's hypotonic would be the outside of the cell in this situation. Okay, so in your box 7, if you could go ahead and explain what hypertonic, isotonic, and hypotonic mean. Now, when we look at this in plants, so hypertonic is going to mean that it's like watering your plant with salt water. Well, now the water is going to leave the cell, and that actually causes, like this area here is the cell membrane, and it actually is pulling away from the cell wall, and this will cause the plant to shrink. That is said to be plasmolysis. Wait, go through like plasmolysis or plasmolysed. Now, plant cells, if they're in isotonic conditions, the water will flow evenly in even rates in and out of the cell. Now, if a plant is in a hypotonic solution, like you water your plant with 100% fresh water, you're going to see that the plant actually becomes very full. That center vacuole fills up with water and the plant becomes turgid. And if you look at the cell membrane compared to the cell wall, you can see right here in an isotonic situation that that cell membrane is not perfectly against that cell wall. And so this plant would kind of be like falling over. Whereas this plant would be standing straight up because that center vacuole is full of water and it's pushing on the walls of the plant, making it turgid. So when we look at this, if you want to think, well, which of these, this yellow part, or what we're looking at, I guess, in part A is going to be a hypotonic or turgid cell. You can kind of see how the cell wall is bulging. That means it's as full as possible and it's pressing on the walls, holding the plant up straight. This one, the plant would be in an isotonic conditions. You can kind of see it's not as firm of a cell wall. And in part C, this plant was watered with salt water or something, and our cell membrane has really pulled away, and this plant would be falling over, unless it has bark on a tree or something. So now I'd like you to write down, what does it mean for a plant to go through plasmolysis, and how does turgid relate to plants? And then lastly, when we look at animal blood cells, when animal blood cells are placed in a hypertonic solution, they're going to shrivel up. So keeping our blood balanced is actually our kidney's job, and we try really hard as animals to keep our blood cells isotonic with our environment. If we drink a lot of water and we actually have the water build up in our blood, creating a hypotonic situation, then our blood cells fill and they'll actually burst. This is called cell lysis. And our blood cells will actually burst open, resulting in death. So a hypertonic solution is bad because our cells shrivel. A hypotonic solution is bad because our cells burst. And for animal cells, a happy isotonic environment is ideal. And so it's kind of like Goldilocks and the Three Bears. This one is just right. Now that's not the case for plants. Plants prefer to be in a turgid or hypotonic environment to press on their cell walls to hold them up. So in your last box, what are the ideal conditions for red blood cells and why?